I want to talk about the problems that are really engaging me right now, in the things that stick in my craw, the things that I sort of am wondering about. And to do this, I want to talk about the work that we're doing in South Africa, uncovering new fossils. I'm going to give you some of the wonderful resources that we've been able to put together to share this stuff with the support of National Geographic, the University of Wisconsin, and some other sources. We have been able to document our work in South Africa from the very beginning on video, audio, and I can, in certain cases, allow people to tell their own stories. So I'm going to start by talking about our discoveries and where we're going with field work. I want to talk then about some of the ways that our discoveries and other discoveries in ancient DNA, in the DNA of modern people, in the fossil record more broadly than our work, are changing our view of human origins. And I want to present some of the things that puzzle me about this, the big problems that I think are facing us now, some of the ways that I think might be promising to tackle those big questions, and then where we're going next. So that's, in a nutshell, where I'm going. This is all the work of many, many people. And I'm privileged to be able to share some of their words with you directly. But I also want to point out that we're working with a team of more than 150 scientists on all aspects of the work that we do in the field. And then all of the discoveries that have happened in other areas of paleoanthropology that I'll talk about are the work of many, many people. And I've got notes on the slides and stuff about whose work this is, but I want you all to know that we are in a field right now that is undergoing a revolution. And you're going to see the evidence of that revolution. And it's the work of all of us together. So first I want to show you Steve Tucker. Steve is one of the cavers who discovered our fossils in the Rising Star Cave in 2013. And he's going to describe to you the first time that he and Rick Hunter, his friend, saw these fossils in the cave.
Caving has always been great. Caving, you want to answer that question of what's around the next corner. That's like always the question you want to know. It's mostly fueled by curiosity. But as you get into caving more and more, the scientific side of it comes into it. You want to know, how did these places form? What happened here millions of years ago? What happened here billions of years ago? Basically, it creates a fascination with this whole environment. In my wildest dreams, I would never have thought that caving would take me to what's happening here. You could almost call this a bit of an accident. So my caving buddy and me, Rick, we were out exploring this cave on a Friday night. We'd gone into a very remote section of the cave, part that I'd never been in before, and in that section, well, we stumbled upon fossils. Yeah, at first we didn't exactly know what fossils yet. We started looking around a bit more until we found a mandible, and that was when we knew this was probably hominid. That was when we got excited about it. And since this discovery, it's crazy what's happening here.
So that was filmed in 2013, and these guys in September of 2013, so fundamentally 5 years ago, were in this cave. They were working with Pedro Boshoff and my friend Lee Berger. Lee is a National Geographic explorer in residence. He's also a prof at the University of Witwatersrand. He's famous for a number of fossil discoveries in South Africa. This cave was totally unexpected. And when I saw the video that you just saw, and when I saw photographs that came out of this cave, I was blown away, right? Because you're looking at an open cave system, very difficult to get into. You see that squeeze. I'm going to show you more about that squeeze that they're doing in just a minute. But, you know, but there they are, right? And it was clear to us from the photographs that this was some sort of non-modern human, some sort of human relative. We didn't know what it was. We didn't know what we were going to find there, but you could see the bones, right? We had every expectation that there was going to be part of a skeleton. And yet, it was in a very difficult and challenging place. The Rising Star Cave is underneath this hillside. It was at the time a disused horse farm, and the cave was used by cavers. It was well known to them because it was a place that had lots of nooks and crannies and squeezes, and it was somewhere that you could get into this kind of interesting stuff without spending a whole day underground. So they used it for training new people. Our cavers that we've worked with now extensively since then, the entire caving society of South Africa, do not get excited by this kind of cave, right? This is actually just kid stuff for them. They get excited by caves called Armageddon, that you have to camp underground to explore, over multi-days, right? So people were not going into these caves with an idea that they're going to find new fossils. And that's despite the fact that we're working in the cradle of humankind area just outside of Johannesburg, where many known fossil sites have been investigated for now more than 80 years. So we knew there were fossils around, but people had an idea of what kind of sites had fossils in them. And this was not their search pattern at all. So this was a surprise, and yet there we were. Now the problem of the cave, I'm showing you here a cross-section of it, of part of it. There are more than 2 kilometers of underground passages in the Rising Star cave system. And the one that's critical to us is this lead in to this area where the bones were found, the Lacetti Chamber. This is a laser scan of the entire cave system. We now have this in 3D, and it's registered against LiDAR scanning of the surface, so we have it in exact orientation with the surface detail. And I want to point out that there are a couple of places that are hard to get through in this cave. One of them is known as the Superman's Crawl, and the reason why is that it's about 10.5 inches in height, and in order to get through, you have to shoulder through it horizontally. But in one stage, the rocks are such that people who are larger, like me, have to one arm forward and one arm back, right? So it's like Superman. This one is a 7.5 inch passage that is a 12-meter vertical climb. We call it the chute. And this was the real constraint. I don't fit in this, Lee doesn't fit in this. No archaeologists that we work with fit in this. In order to investigate what these bones could be and to figure out this site, we had to recruit a special group of people. And so Lee went on Facebook and said, I need help. Please send this message out to as many people as possible. We need people with archaeological experience who have caving or climbing experience. The catch is this: the person must be skinny and small. They must not be claustrophobic. And they have to be available next month. 57 people applied for this. And Lee was able to select a team of all archaeology PhD students from the US, from Canada, from Australia, and this became our underground expedition. They were supported by a series of volunteers, mostly from the Caving Society, but including some people from Wits University and public. We had 60 people on site to support this team underground for a month in November of 2013. And including me, right? I gotta tell you, I like showing you guys this slide to let you know that I do go into caves, but the fact is I'm useless. It's like I say, right? This is a team enterprise, and I have certain abilities that are really useful for this whole exercise, but going into skinny places underground is not among them. I will say, right, we have a second fossil site in the cave, the Lacetti Chamber. I'll show you the fossils in a minute. But the Lacetti Chamber is a little bit easier to get to, but I still can't make it. And Lee went there once. And on his way back out, he was stuck in the squeeze. And the squeeze is not a crazy, crazy squeeze, but when I saw him go through it going down, I was like, well, I guess I'm stuck here, right? I can't fit through that. And when he came back up, he didn't fit. And so it was literally 45 minutes of getting him through the squeeze. That squeeze has become known as the burger box. So there's, we have fortunately, in the time that we've been working there, not had any serious safety events. And I'm going to show you the chute. This is Rick and Megan Berger actually in the chute. I'm going to show you what this is like for our team coming up. So what you're going to see to start out with is And I'm going to turn down the program volume because we'll need that. Actually, mute the program. What you're seeing here is Becca Peixoto, who is coming up the lower part of the chute, and Marina Elliott has got my Steadicam. She's about to hand this camera over to Becca, and you're going to see a camera switch. Becca's squeezing into this little crack that actually Marina, when she saw this video, said, where is that? I don't even know where that is. Because the different team members who are different heights fit in different ways in this passage. So here is Marina now taking the next section of this. And you get a real picture of the physicality of this, the athleticism that all the team has to be able to do this. I want to tell you that everything that we do in this chamber, all the equipment that we use, all the people who access this have to be able to make this climb. And as you can see, it's a free climb. Safety harnesses actually impede the team if they're going up and down. They're safer without a harness on. And so everything that is in the chamber eventually has to come back up. That includes lighting, wireless routers, computers, and the fossils. And so this place takes our team members who are incredibly skilled underground. This is Ellen Feuerriegel, who's taking now the top part of the chute. Takes our team a half an hour to traverse through the cave into the Lacetti Chamber to work, and a half an hour to come back out. So it is really, really a difficult place to work. We've likened this to we call them underground astronauts. And the reason for that is their only communications when they're in the chamber are virtual. You know, we're able to talk with them on telephones, actually, but there's not any ability there. It's like a spacewalk. You know, you really are in a remote place that you can't, get help immediately if you need it. Okay, so a month out underground, our team working above ground conserved the fossils as we went, and over the course of that first 21 days underground and a second field season of more limited scope, 13 days, we recovered more than 1,500 fossil specimens of what we really quickly discovered was a previously unknown species. We named it Homo Naledi. I'm going to tell you a bit about Naledi. I'll give you an introduction. We've recovered every bone in the skeleton, at least in partial form. And we've recovered bones from a minimum of, in the initial phase, 15 individuals. Today I can increase that. We have at least 20 individuals in the Lacetti Chamber. They represent each body part, and most of them multiple times. So we have a very good record of the anatomy of the species. I'll show you some salient features of it. First, lots and lots of cranial remains, and this is true of most paleontological sites. We find lots of skulls and teeth, and the skulls and teeth are very important. I'll talk about why they're important in a bit. But with Naledi, the biggest, most obvious thing is that their skulls are very small compared to our skulls. They have brains about 450 to around 600 cubic centimeters in the very biggest individual we have. That is about a third the size of your brains. So we're looking at a very small-brained hominid. This is John Gurchi's reconstruction of what Naledi would have looked like when it was alive, and I think this is, I love his work. I think this is as good as an artist can do. Obviously, some aspects of this are more speculative, but they're still based on some sort of scientific point of view. Skin pigmentation, for instance. We don't have any bony knowledge of this, and so we use what we know about the modern human distribution of skin pigment. The brains, I'll talk more about brain anatomy in a bit, but we do know quite a lot about the brain anatomy externally of Naledi. We have the remains of at least now 20 individuals. I will tell you that we have individuals of all ages represented in this sample. This is a very old adult. You can see here the wear on the teeth has gone down to the roots on the first molar, so we're looking at somebody near the end of their lifespan. Here's a toddler age individual, about a 2-year-old if they aged in human terms. I'll tell you, we don't know precisely how fast they developed, how fast they aged. chimpanzees develop much faster than human children do. And so we don't know exactly where Naledi lies, but their canine eruption is late, their canine eruption is early. It's late in chimpanzees, and so there's a hint that maybe this is a more human-like pattern. Their teeth are around the same size as your teeth. Their incisors in the front are a bit smaller. Their premolars are about the same size as yours. Their first molar is just a hair bigger than your first molar, depending on how big yours is, right? Some of you are big tooth, and so this would be maybe your size. The second molar and then the third molar, the wisdom teeth are not in this individual, are bigger, and that's very different from you. Your third molars, if you have them, how many guys have third molars, wisdom teeth? Yeah, they're smaller than your second molars. If your third molar is bigger than your second molar, we want to see you. It's not impossible, right? But it's rare. Your second molar is smaller than your first molar. For Naledi, it's the opposite, and that's a primitive pattern. That's something that we see in early hominins like Australopithecus, like Lucy. We don't see it in living people. Their premolars have a very primitive morphology as well. You don't see, especially this third premolar, you don't see that anatomy in any living people. It's in fact quite unique. We don't see it in Lucy either. Their third molars are large and highly asymmetrical. This is very symmetrical too. We can look at the rest of the skeleton though, and that's actually quite rare. Skeletons in the fossil record, we've got a lot of evidence about human origins from fossils. A lot of that is, however, biased towards skulls and teeth. And we have postcranial bones of many species, and we have some partial skeletons, some really, really cool ones. What we don't have for most species other than modern humans and Neanderthals is a representation of the skeleton of one or multiple individuals that lets us correlate parts with each other. And so this becomes a really important sample. I want to talk about some of those parts. This is in the site, as it was unearthed, a hand. You can see here the metacarpals, the carpals of the wrist. You see here finger bones. These are the middle phalanges, the intermediate ones that are curled over. Right? This is a hand in a death pose. In articulation in the site. And this is that hand. Here's the palm, here's the back of the hand. I want to show you a couple of things. The finger bones, the distal ones, have really broad fingertips. That's like us. Chimpanzees have super narrow fingertips, as does Lucy, as does any kind of early hominid. Big thumbs, stout thumbs, wrist bones that are configured like ours. But I want to show you the finger bones are really curved. And that's not like us at all. Our finger bones are very flat. And curvature is something that occurs during development as the growth plates are activated on a curved substrate. A curvature can emerge. And that's what we're looking at here. We're looking at hands that were used to grasp onto curved things. The thumb also, I want to point out, has got this, there's a couple osteologists in the room, right, who really know thumb bones. And I want to show you, the base of this is really small, and the end of this, the distal end, is really large, and there are these huge ridges on it for the musculature of the hand. And you look at that and say, wow, that's really impressive, right? That must be some strange thing about this individual. And this is the fossil site where I can show you 7. And I can show you that then they all have the same kind of morphology. We're really looking at a population that has some odd things in it that we haven't seen before in the fossil record. Feet are human. My student, Zach Throckmorton, who's now an anatomy professor at the Northeastern Arkansas College of Osteopathic Medicine, he came to study the feet, and he was looking at these foot bones. And after a few days of studying the 107 foot bones, right, he says, "Why did you bring me here?" And I said, "What do you mean? Isn't this amazing?" You know, he says, "These feet are completely unremarkable." And I said, "You're kidding me." Right? He says, "These could have walked into my dad's practice." Now, his dad was a podiatrist. And I've got to tell you, Zach knows everything there is to know about feet, right? He knows it all, right? And so when I hear him saying this, I'm sort of concerned, you know, because I think these are cool, but I don't know feet like he does. I had to pull him over to the next table where they're working on the skull, And I said, did that skull walk into your dad's practice? Because we're looking at something that has human-like morphology, very human morphology in its lower limb, and has a very primitive skull. And that's really weird. Lots of feet from the site. If we look at what Naledi looks like as a figure, right? This is, again, John Gurchi's painting of this. I want to show you, this is Homo erectus. This is a skeleton that we know called the Nariocotomy boy, or the Turkana boy. This is Lucy as a reconstruction, right? And I don't go too much into the hair and that sort of thing. I just want to show you, Lucy's got these big shoulders, she's short, she's small body mass, she's got short legs. Turkana boy has a very human frame. You know, his proportions are fundamentally human-like, even though his skull isn't exactly ours. Naledi is this weird combination. Shoulders that are sort of canted upwards, very muscular, but not super long arms, legs that are proportioned very much like ours, a very small skull, right? This is a strange mixture of characters that we haven't seen before. And so we're faced with a problem. How do we explain this? How do we explain the presence of many, many individuals of this species in a deep cave chamber with no other animal bones other than 6 bones from an owl? It's weird. Well, almost immediately, as we were working underground in our first phase, our cavers brought to us a piece of news. There is another place in the cave where there are bones. Our chamber, the Naledi Chamber, this is an overhead view of the cave system. The Naledi Chamber is here in yellow. Our surface entrance is here in blue. This is where I sort of hang out. And this pink area is called the Lacetti Chamber. The Lacetti Chamber, produced a partial skeleton, which was identical in its morphology to the first chamber's skeletal material. This is an individual, we've nicknamed him Neo, which means gift in Sesotho. Naledi means star in Sesotho. So we have an individual there, along with a couple of pieces of other individuals, that's showing that Naledi is in this cave system and using some significant parts of it. I'll give you one other piece of information. We didn't initially know how old Naledi was. After 3 years of dating methodologies, giving samples to multiple independent laboratories blind, so that they didn't know what they were looking at, right? And they didn't know that 2 different labs had the same sample. We derived an age estimate for the Naledi Chamber fossils. They are between 236,000 and 335,000 years old. These fossils, if you know anything about human evolution, you know Lucy is 3.5 million years old. Our evolutionary history is 7 million years, and these very early small-brained creatures come from the early phases of our evolutionary history. Never have we found a fossil younger than 2 million years in Africa that has morphology like we're seeing it. This fossil species lived at the same time that modern humans were originating in Africa. All right, so that's a big story. What I want to do is probe. I was really privileged to lead the description of Neo, and these photos won one of the top 10 science images of the year last year, so I was really, really pleased about this. Let me talk broadly about human evolution. This picture, the famous March of Progress, is not how it happened. This was from a Time Life series in 1964, and it's a wonderful piece of art. It's really iconic. People have repeated it again and again. Google, for instance. You see this everywhere, and we all know this is not how evolution happened. And yet the image is burned in our minds. I want to talk about what we thought about human evolution 15 years ago. Okay? And there's a diversity of opinions, and I'm going to represent a bit of that diversity, but I want you to know I'm stereotyping a bit, but I want to focus on some issues that really represent new information. All right, so here's a picture of brain size over time, right? This is time 3 million years ago, 2 million years ago, 1 million years ago today. Brain size 400, 600, 800 cubic centimeters. You can see that there's an early phase of our evolution, which we call Australopithecus, that is small brains. And there's a later phase that we call Homo that represents brain size increase. And it has been somewhat, this is a log scale, right? So it's actually curvilinear. It accelerates a bit as you get toward the present. That's 15 years ago. 15 years ago, we knew that the March of Progress image was wrong, right? If you read Human Evolution 15 years ago, they're talking about how bushy our evolution is. But I've drawn a couple of pictures from that era just to let you know, when they're talking about bushy, they were talking mostly about this middle part of the tree. This part that represents the last Australopithecines, including robust Australopithecines, and the earliest members of our genus Homo. And as you get toward the present, they realized that there are multiple populations here, but they tended to draw sort of straight lines between them. You know, like something about our early evolution was really speciose, diverse, and our later evolution became a bit more focused. Like there was a trend. And that trend was manifested maybe differently in different regions, but the trend was real. There was a lot of disagreement about what happened to certain kinds of archaic humans. This is a Neanderthal skull. This is from Lacetti, France. This is a Cro-Magnon. You know why I call him a Cro-Magnon? Because he's from Cro-Magnon, also in France, right? This is about 30,000 years old. This Neanderthal about 50,000 years old. This skull is an early skull from China. This is from Liuzhang, China, and it's about 65,000 years old. All right, so we're looking at here two skulls that are about the same age, maybe this one a little younger, and this one after the Neanderthals. There was a lot of disagreement about what role Neanderthals might have had in our origins. The idea that modern people throughout the world had an African origin was especially noticed and became a big subject in the 1980s. So this wasn't new information 15 years ago, but there was a debate about the role of growth of an African population. Did they replace all of the Neanderthals and other kinds of early people, or was there some kind of interaction between them? Do people have Neanderthal genetics today? Right? There's a lot of disagreement about that 15 years ago. I would say we all knew that Africa was the focus of our evolution and that African populations had spread. The question was whether there was an elimination of Neanderthals and other things, right? So that was 15 years ago. I would say, however, that everyone in the world who was an anthropologist thought that if anybody has Neanderthal DNA, it's going to be these people, who were Europeans after all, and not these people, who were Chinese. And certainly not Africans, right? So the idea was that there was a regional contribution possibly, but there might have been a complete replacement of the Neanderthals. That's 15 years ago. Now this opinion was not universal 15 years ago, but it's representative of an opinion that was respectable 15 years ago. All right, Stephen Jay Gould, very famous biologist, writing in about 2001, so this is before his death. There's been no biological change in humans in 40 or 50,000 years. Everything we call culture and civilization we've built with the same body and brain. All right, now some of you in the room are evolutionary biologists, and you're like, how could that be possible, right? We all evolved. Oh no, we evolved. Let me just say, 15 years ago, I was publishing on recent human evolution, and I was really facing an uphill battle. And this was a really respectable opinion, that what's different about people today is cultural. There's nothing about us that has changed in any massive way that's biological. And another thing, this is now 20 years old, but it was a respectable opinion also 15 years ago. Eric Lander, most genes have only a handful of common variants in their coding regions, with the vast majority of alleles being exceedingly rare. That's true, right? That's the allelic spectrum of variation in humans. This limited diversity reflects the fact that modern humans are descended from a relatively small population that underwent exponential expansion in evolutionary recent time. This is also true. This spectrum of frequencies of alleles comes from our demographic growth. Tantalizing examples suggest that common variants may hold the secret to many disease susceptibilities. This was known as the common disease, common variant hypothesis. The idea that when you look at traits in human populations, common variants that are common in human populations will account for a substantial degree of the explanation for why the trait is heritable. This went behind the funding of the Human Genome Project, right? This was why we spent so much to sequence the genome, because we're going to find out what causes disease. 15 years ago, these are biological sort of ideas about humans and their evolution. Okay. Now, let me run through what's happened in 15 years. First, we have ancient DNA sequences from ancient humans. We now know what the contribution of Neanderthals was to human populations, right? Here are Neanderthals. They branch off from modern humans sometime in the distant past. This is Middle Pleistocene age, maybe 500,000, 600,000 years ago. And there's a contribution of Neanderthals both to the early evolution of humans and, here's another chart, human populations in different parts of the world, Neanderthals, and from an early branch of modern humans into some Neanderthals, and from some other Neanderthals into other later people. In addition to that, there's another branch, a branch that we've discovered at a place in Siberia called Denisova Cave, and this branch represents a very early branching of this archaic human Eurasian lineage, so different that researchers have decided that this isn't just a Neanderthal, that it represents a very deep population. They've called it Denisovans. That population also contributed some of its genetic material to living people, but living people primarily in Sahul. That's Papua New Guinea, or New Guinea broadly, right? Irian Jaya as well. And Aboriginal Australians. And a tiny fraction of their DNA to people in East Asia. So there's multiple contributions from archaic humans from their genetics into living people. All right, so we now have settled that. We know what the contribution was. We have found hybrids. Just a few weeks ago, this nature cover, right, represents an individual from Denisova Cave who has part Neanderthal and part Denisovan. And you'd say, well, maybe that's just a mixture, right? That's very much a mixture, because this individual, every chromosome position where he has one Denisovan variant, she has another chromosome position that's a Neanderthal variant. So in other words, every time you've got one that's like a Neanderthal and one that's like a Denisovan, that's an F1 hybrid. So we've found hybrids of ancient people. It's not just that they hybridized with us and introgressed their genes into us, which they did, but also we've found actual individuals who represent the direct mixture of them. This is not so weird. First, I want to show you a couple of charts that show that humans are not so weird. For instance, these are chimpanzees and bonobos. You have these multiple subspecies of chimpanzees today, eastern, central, western chimpanzees, and bonobos. They also have exchanged genetics in their prehistory. Right? What's weird about humans is not the fact that we had these genetic exchanges with past people. What's weird about us is that the past people aren't here. And so today, we're really uniquely uniform. Here's an elephant phylogeny. We're a lot more like the elephant case today. You've got mammoths, Columbian mammoths, woolly mammoths. You've got an American mastodon here. The mastodon doesn't mix with these others. It's quite distantly related. They separated like 20 million years ago. But the elephant phylogeny, which started about 5 million years ago and separates African elephants, that's Loxodon africana today, from mammoths and Elephas maximus, that's Indian elephants, and all of these paleoloxodons, they've all mixed with each other. Over 5 million years of mixing. So, yeah. And the elephant phylogeny is a bit like ours in that there's only a handful of these representatives left, right? The mammoths are gone. Indian elephants and the 2 African elephants are all that remain. So, wow. You know, there's nothing unusual about our evolutionary history and the extent of mixture of populations. What's unusual is that humans today are the only ones left. This was not a new idea, right? This is a model from Jeff Wall in his lab in 2009. Right? So I don't want to say it's ancient DNA that made this happen. We have signs from living people's DNA of diversity that was difficult to explain from just one very fast-growing population. And it's in part that's given rise to the next big thing. But the mixture that remains is fairly consistent at a low absolute proportion of everybody's total ancestry. So this is Neanderthal, no, this is Denisovan genetics throughout the world. And you see here it's high in Australia, New Guinea, very low in East Asia, and pretty much 0 everywhere else. Neanderthals have a consistently low fraction that ranges up to about 3%, and is never higher than that in living people. Okay, I'll skip this and go straight to now. I showed you this one before, and you might have noticed potential unknown hominid. Once you have ancient genomes, and you know that ancient genomes contributed to living people's genomes, you start looking at that closely and you start to realize, wait a minute, that looks like a pattern. I know what an ancient genome looks like when it mixes in with a living person's genome. And once you know what pattern to look for, you can look for it without the ancient genome. You could say, let's look for the footprints of ancient mixture in living people's DNA. We have found those footprints, not only in living people, but also in ancient genomes. This potential unknown hominin is the first of the ghost populations that's been identified. It diverged from everybody else that we have ancient genes and living genes from sometime around 1 million years ago, way before Neanderthals and Denisovans branched from each other, and contributed a fraction of the ancestry of the Denisovans. So this Denisovan genome, which I've already told you is strange, is doubly strange because it's got some input from somebody mysterious. We don't know who. Here's that Denisova finger. Finding things that we didn't know were there has been a common feature of my field in the last 15 years. I want to give you some examples. Denisova is a big example. The genome that has identified this ancient population came from this bone. Wow, that's not much. An archaeologist would not have made anything out of this. You know, this is the sort of bone that goes in the cabinet of curiosities. Oh yeah, we've got a hominin phalanx. Want to see? What can I learn from this? Nothing. It's an immature. You've got a growth line on here. That's all you could learn from it. Until genetics came along, and it was like, oh yeah, there's a whole genome here. It's something we've never seen before. Wow, you know, that's amazing. But it doesn't take genetics to do that. This individual, this may be the earliest modern human that's ever been discovered outside of Africa. This maxilla comes from a place called Mislia Cave. It's around 180,000 years old, and it's in Israel. It's way older than it ought to be. We've never found modern humans outside of Africa that early. Wow. This skeleton, this is from a cave called Liangbois. It is on the island of Flores. It has a very small skull. It has strange looking bones. It has a small stature. It has very primitive feet and wrists. It looks really different from modern humans. It lived there until 60,000 years ago on an island that it had to get to by crossing 2 deep water straits. That was discovered in 2003. That's why I've chosen 15 years arbitrarily, because this one, these start to show up. Homo Naledi, right? Here we go. Here's something we discovered. We had no reason to answer. You guys could look at Flores and say, Flores is an island. Weird things happen on islands. Here's something strange. that we can integrate that easily into our view of human evolution. Strange things on islands, that's not a big deal. Strange things in the middle of Africa are a big deal. This is a continent on which our species was originating and that was home to large-brained human relatives throughout the last 2 million years. And here, 250,000 years ago, we found something very different. That's weird. And let me just note a couple of things. I've shown you this one before. I just want to say, this guy, Cro-Magnon, 15 years ago, all anthropologists would agree that this was an early representative of today's European population, that this was an ancestor of today's Europeans. It is an ancestor, but this individual's population, the early Upper Paleolithic population of Europe, represents less than 10% of today's European ancestry. This is almost a ghost population. We know that in part because we have other individuals, this one from Oase in Romania, this individual is 10% Neanderthal. Nobody today has anything like that. There was a mixture that happened in Europe that predates today's European population. Today's Europeans mostly derive from somewhere else. And I say most, of course we mostly derive from Africa regardless, but not only that, we also mostly derive from somewhere not Europe within the last few thousand years. And when you look at the DNA of these ancient Europeans, you start to see, here's the DNA distribution of populations today in Eurasia. You see this range of variation. When you look at early European farmers, that's 6,000 years ago in Europe, they cluster with a population. This population is Sardinia. Farmers from Northern Europe, where today they should be up here with Sardinia. And those early Cro-Magnon type hunter-gatherers, they don't look like anybody living now. So that when we reconstruct these relationships, this is a program called Tree Mix that gives rise to this formation of a tree with dotted lines that represent mixture and ancestry between groups. We reconstruct these ancient populations that we don't have representatives of. These are ghosts. We don't know who they are. And that seems to be a recurrent feature of our field in the last 15 years. We find things that we didn't know existed before, and we now can predict that things existed that we haven't found. Okay. Yeah. Okay, so our evolutionary history, I like this analogy a lot, was like a braided stream. During the last million years or more, we have had very deeply diverged populations that sometimes encountered each other and sometimes mixed with each other. It doesn't look very much like today's human population, but it does look a lot like the populations that we see of other kinds of large mammals with large geographic ranges. There's nothing really weird about us, but it's a different analogy than we're used to thinking of. Now, I want to talk about a couple of genetic things that bother me. I'm going back to this chart. One thing that's true is that these people that were ancient people that don't look like living people, you might ask, why are they different from us? And one reason is that natural selection has been affecting all of us. Natural selection doesn't affect very many genes in a genome-wide comparison, but it affects some genes a lot, and those changes have been really noticeable. This is frequencies in ancient population samples of the allele that today lets people digest milk. This is lactase persistence. And you see in the most ancient samples, it is 0. Ancient people living today in Germany, in Norway, in places that today people digest milk quite easily because the frequency of this is very high. 8,000 years ago, they didn't have it. This is new. And it increased over time. This is a model of how natural selection should increase an allele over time. It increased over time in the way that selection increases alleles over time. We were selected. It was lots of evidence of selection recently. This is some of my work from 2007, and we looked at variants that looked like they could be selected, and we said, wow, this is very strange. If you projected this back to see what should be fixed in our evolution, it would be a very, very large number. We should have evolved super fast. And we didn't evolve super fast. It just looks like we're evolving recently kind of fast. And that's interesting. But more to the point, when we look at most traits are not traits like lactase, where one gene makes a big difference. Most traits are polygenic traits. They're traits that many, many genes make a difference. And in the last 5 years, and especially even in the last 1 or 2 years, it's become possible to look across many, many, many genes and make a score, what's called a polygenic score, predicting what somebody's phenotype should be based on the heritability that's spread across hundreds or even in some cases thousands of gene loci. And when you do that, this is stature. This is how tall your genes predict you should be and how tall your genes are. So these are populations of humans, and you can see that there's a good agreement of this, and that agreement constitutes a cline. There's a cline. These are West Asian populations, North Asians, East Asian, South Central Asian populations. There's a cline over space that corresponds to a cline of polygenic height. That's selection. That's selection on height in these populations. Now, these polygenic situations, we're just now getting a grip on, right? When I introduced at the beginning, Eric Lander, the common disease, common variant, the idea that common genes will explain a lot. Common genes do explain a lot, but you have to look at lots and lots and lots of common genes to explain. And looking across lots and lots of genes, every trait that's a disease-related trait, and disease I'm pointing at because people study disease a lot, not because they're unique, has these lots and lots of alleles that can relate to it. These are called complex traits. It's opposed to a simple trait. And some diseases are simple traits, like hemophilia is simple in the sense that one gene change can cause it. It can be complex in the sense that most of those are rare, and there's lots of rare ones. Today, when we look at heritability across traits like that, we think about it in terms of models. And one common model today is called the Omni Genic model. I want to point to this because the insight, and this is attributable to Jonathan Pritchard and his students. The insight of this is that even if most genetic changes are really due to changes in a small network of genes that really, really are functionally connected to a trait, that network of genes is co-expressed in different kinds of cells with other genes. And that co-expression actually influences how the core genes are expressed in the cell. So as a consequence, heritability becomes smeared over lots and lots and lots of genes. That's a really core insight about the way that human genetics is influencing our traits. And the reason why I bring it up, it's an insight from human genetics that I'm bringing in. The reason why I bring it up is because of this problem. Remember I said the Neanderthals, everybody agreed they contributed to this population if they contributed to anybody. And everybody agreed that they wouldn't have contributed to this population, the Chinese population. Chinese people today have more Neanderthal ancestry than European people today. This, to me, is a deep problem because every anthropologist 5 years ago, right, well, 8 years ago, would have said, the Neanderthals, if they contributed to anybody, contributed to Europeans. Why? Well, Europeans share some features with Neanderthals. There's some features that they share. People who argued against that would say those features are shared by convergence or parallelism. They're not meaningful in terms of genetic interchange. They're just because they live in the same place. Today we appreciate that Neanderthals lived not only in Europe, like France, but also across most of Central Asia. They were broad in their population distribution. And today, Chinese people have more Neanderthal than Europeans. That suggests, why do I think this is a deep problem? That suggests that until 8 years ago, when we had genetic information from these guys, my entire field was barking up the wrong tree. And that's a problem, right? Because the only way that we have to study every other fossil is by looking at what they look like. Let me show you. Homo naledi, here's its brain size. This is brain size. This is naledi. Here's Homo floresiensis, right, the hobbit. Here's some Australopithecines. Here's Homo habilis, bigger than Naledi. Rudolfensis erectus, bigger than Naledi. If I put Naledi and Flores on this same chart that I showed you a bit ago about brain size, you see brain size gradually increases. Wait a minute. What is the deal here? There are these things we were missing. And yet, when we study, and our work earlier this year came out in PNAS, when we study the morphology of Naledi brains externally, they look very human-like in the aspects we can see. Right? They look like Homo brains, even though they're smaller than any other Homo brains. Okay. You look at Naledi and you say, this looks like it should be like these primitive things and not these more progressively more human-like things, right? It really does. And that's also true of the skeleton. Here's traits, and you can see that Naledi often resembles these primitive early hominin species and rarely but occasionally resembles modern humans and not those other species. If we do a cladogram of this and say, where does Naledi fit? Looking with, some of you will get into the details of this, and I don't want to go deep into the weeds, but I do want to say, I got 2 cladograms to show you, done with the best methods of 2 different types. This is a Bayesian tree, and the other one is a maximum parsimony tree. And they put Naledi in totally different places. When I look at the fossil record, I want to know how species are related to each other. I'm going to use their morphology because that's what I got. And I've just shown you that genetics shows that what we thought about the fossils we know the most about, Neanderthals, was wrong. And now I'm showing you that these other fossils that we've recently found, the morphology puts them in totally different places. It's weird. It's wrong. It's disturbing. Our tree was bushy, and what's interesting about this is not per se the bushiness. We've known that a long time. What's interesting is that these question marks are really intractable question marks. We don't have evidence today that tells us with any authority how these species are related to each other. So I want to turn to the evolution of our human ancestors in Africa. And I'll tell you, we have some fossils of early modern humans. They're highly diverse. They seem too variable to be one very tight population. They're diverse. And some of the population structure that genetics shows us exists today in Africa, between Khoisan people in particular and other Africans, dates to more than 300,000 years ago in terms of population structure. So that early modern human population was diverse. It was already divided into multiple populations at the time that Naledi lived. Naledi lived among our diversifying species. And signatures within African populations show that there were contributions in Africa from groups as different from each other as Neanderthals and Denisovans are from us. There were ghost populations in Africa. We've got fossils from almost the Holocene. This is 17,000 years old. This is from Iwo Eleru in Nigeria, that have a really not very current human-looking cast to them. They might represent these ghost populations, or something like Homo Naledi might represent this ghost population. We don't know. This is Naledi next to one of the earliest modern human skulls. This is from southern Ethiopia. These two could have lived at the same time. These these tools from Katupan are 500,000 years old from South Africa. And more broadly, the tools across Africa found in this time frame when Naledi lived are what we call Middle Stone Age tools. They're advanced tools. They're the tools that we think go along with modern humans. How did this happen? The answer to this question is, I don't know how it happened. We're going back in the cave to find out. We're still digging. We're trying to uncover evidence that would relate cultural evidence to Naledi. We've gone back into the Lacetti Chamber. We're excavating now more areas. We're uncovering more bones. We've found bones in unexpected places. And our team underground is just doing amazing things. They're back in the field this month. I want to show you another creepy video, and this is just to end. All right? Here's our team in the back sections, the fissures that lead off of the Lacetti Chamber, where we have found bone remains of Homo Naledi. This is Steve Tucker. You saw him at the beginning. He's about to take off his helmet here because his head doesn't fit with his helmet on. I don't know the answers to these big questions in my mind. They puzzle me. They really bother me. I know that the way that we're going to find the answers is with more field work, with more exploration, finding other caves like this. And we have recovered in the Rising Star Cave this year a skeleton of an infant that we have in a plaster jacket in the laboratory, that has a complete dentition, that has all of the information skeletally that we're going to dissect virtually. Nobody will touch it. So the hard work in the field goes on. I think we're going to find the answers to some of these things, but these are big questions. They deserve us to think about the past in a new way, to try to understand how to bring to bear all the evidence that we have from different sources. And that's what we're doing with our large team, and that's the way that I think the field is going to change. Okay, I've gone a little over, and I just want to be respectful of everyone. If you have to leave, please don't think you're offending me. But I'm also willing to stick around for a little bit and answer questions if you have them, so please feel free. And with that, I'll just say thank you again so much for your attention and for coming.